it's true, science saves lives, it transforms our universe, it can even make you a fortune if you're lucky, but that's not why we scientists do it. We do it because we're simply curious, and it's so exciting to find out something new, to be the first person in the world to see it and to understand what's going on. And so it's a purely selfish reason that we do it. But sometimes, as has happened in my life, you're incredibly lucky, and it does help other people in the long run. And so what I will try to do in this little brief talk is tell you a few things about science and why I think science is so important, and the things that you have to think about if you're doing it, and then tell you a little bit about my own work and how it's helped people with a very rare form of diabetes to be able to throw away their insulin pens and take a drug twice a day instead. So the thing that sustains me throughout the very long hours in the lab and 30 years of work is the thrill of discovery. And when I first arrived in Oxford, I decided that what I really wanted to understand was how glucose causes, how rise in your blood sugar concentration causes the release of the hormone insulin from the beta cells in the pancreas. And most of you probably know that insulin regulates the blood sugar level, and that if you don't have enough, then you will die within a few minutes because the brain is starved of fuel, and that if you've got too much for too long, then you will get all the secondary complications of diabetes. So I set out to try to understand how a rise in your blood sugar causes the release of insulin. And I had the idea that what might be going on was that a tiny pore that's found in the membrane of every cell of your body, this family of proteins is found in every cell of your body, might actually be going wrong. And so I set out to look for this. And for those of you who are medical students, what I'm talking about is an ion channel. And this ion channel is very important for regulating the electrical activity of the beta cell. And what happens, and what I discovered, was that what glucose does is to shut this tiny pore. And the consequence of that is that insulin is released. And I'll just tell you how wonderful it was that night when I first saw that happen. So I'd been working on this for some time and nothing had been working, and then one night, at about 8 o'clock at night, suddenly I saw that when I perfused the solution with glucose in it across the cell, this tiny little pore shut, and the current that I was recording that flows through the ionic current disappeared. And I was really very excited, and I thought, well, you know, what this must mean is this is how glucose stimulates insulin secretion. This must be how all the drugs which are used to treat adult onset diabetes work. This must be what goes wrong in diabetes. And then I thought, well, maybe this is not right. Maybe this is just an experiment that's gone wrong, you know, some disaster in the electronics, which often happens. And so I realized that the only thing to do was to take the glucose away again and see if the channel activity came back. And when it did, I was over the moon. I was so excited. Unless this happened to you, you simply don't know the thrill of discovery. It's better than anything else. As one of my friends said, it's far better than sex, and it lasts for longer. And so I was really excited. I went home, I couldn't sleep, I couldn't eat, I was just, wandering around. And then, sorry, can you hear? Is it not coming? And then, by the next morning, I decided that everything was nothing more than rubbish. It was just a mistake, and I made an error in something or other. So I came into the lab, and one of the people I'd run the night before in my excitement came in, and I said to him, well, I think it's all rubbish. And he looked at me and he said, Fran, all you have to do is repeat the experiment. Because of course, in science, that's what you do. You do an experiment, you get a result, and then you make sure that it happens every time you do the experiment. So that's the other side of the science. from the exhilaration of discovery to the daily drudgery of doing the same experiment again and again and again for many years. And when it doesn't work, doing it for year after year after year. But you see, the thing is, if you're interested in what you're doing, there are all these small little discoveries, these small little things that happen as you go along, sustain you until you are able to answer eventually by putting all these bits together, the bigger picture. So that night I felt like Stout Cortes on his peak of Darien, looking out across a new landscape, an intellectual landscape, knowing where I was going to go for the next, as it turned out, 30 years. And it took a long time to get there, because many, many things had to happen along the way, such as we had to clone the channel and get its DNA sequence. We had to sequence the DNA from people with type 2 diabetes. And when we did so, we didn't find any mutations. We found no genetic defects that were associated with the disease in this protein. And so I'm afraid I couldn't believe that was the right answer. I was certain they must be there. And I realized, well, one of my brilliant postdocs pointed out that we were looking at the wrong people. We should be looking at people who were born with diabetes. And I went and I asked all my clinical colleagues, can we have some DNA from people who are born with diabetes, please? And they said, oh, those people don't exist. So I foolishly believed them. So to those of you who are going to be scientists or medical students, just let me say, follow the motto of the Royal Society, nullius in verbum. Take nobody's word for it, because those people do exist. They're just very rare. And 10 years later, a colleague of mine rang me up, and another second exciting day in my life, when he said, Fran, I think you better sit down. I've got something really exciting to tell you. And he told me they'd found those first mutations. So why was this exciting? It was exciting because it told us that these mutations in the gene I've been working on caused people to have diabetes at birth, a very rare disease. And this actually was transformative in my life. Normally, if you're a scientist, you never expect that the work that you do will have any application in the wide world until many, many, many years after your death. And that's usually what happens. But I've been incredibly lucky because it's happened to me in my lifetime. Because what we realized was that the people who were born with diabetes had been treated for many years by insulin injections. And what we knew from our studies was that the problem was not that they didn't have any cells that released insulin, but that they weren't able to release it because these tiny pores were open. So what we could do was give them a drug that shut the pore, and we had that drug. It was used in adult type 2 diabetes. So we then had to, this was not me, this was my clinical colleague, Andrew Hattersley, we had to then take people who were being kept alive by insulin injections and say to them, we think we might have a better treatment for you. We think you could give up having injections several times a day, and you could just take a pill instead. But actually, we don't really know that. We have to do the experiment, because it's possible you haven't got any beta cells left. But we think you probably have, but would you mind having a go and taking this pill? And one brave woman in the Netherlands said, yes, I'll have a go. And it worked. And now several hundreds of people, at least 1,000 I think, have now moved from insulin injections to taking a pill. And you can imagine that if you're a mother born with a tiny baby who's just a few weeks old and they require insulin injections, which causes them to scream, and you have to get up every few hours throughout the night either to feed them or give them their insulin, that to be able to just give them a drug ground up in their milk is so much better. And for me, the thing which fascinated me was I had not realized how liberating it would be for people who are in their teens or in their 20s and 30s to switch, because some of them said the most extraordinary things to me. I think for me, the most emotionally rewarding thing was the young girl who said to me, very beautiful young girl, very slim, and she said, you know, thanks to you I can wear a dress. And I said, what do you mean? she said, well, I always used to have a waistband to hang my insulin pump on, so it always had to be a skirt or trousers. But now I can wear a tight-fitting dress because I don't need it anymore. And I thought, you know, what a wonderful thing. And so I have been immensely privileged and immensely lucky. And I have also been extraordinarily lucky in that I've been able to meet the people who our work has helped, because we decided we'd run a conference for them because it's such a rare disease. So we invited them to London, and the Wellcome Trust paid for it all, and the Royal Society hosted it, and I got to meet these people who are now personal friends. And amazingly, one of them was here yesterday and the day before making a film about this whole story, and came to England to interview Andrew and myself. So this has been an incredible journey. It's also been a long journey. Never imagine that science is easy. Science is tough. Experiments don't work lots of the time. Things go wrong. But the addiction of discovering something new is just so all-consuming that actually you can't stop. And the challenge of trying to answer that question means that you try it again and again and again. And I think you should listen to the words of John Kennedy, what he said about putting a man on the moon. We do these things not because they are easy, but because they are hard. Because that goal will serve to organise and measure the very best of our energies and skills. Because the challenge is one that we are willing to accept, and one that we intend to win. And of course, if you put your mind to it, and you work hard, and you carry on for a long time, you will get there. Nothing is ever impossible. Now, you might also say that science can be boring, because you need to repeat the experiment many, many times. But, you know, you're always buoyed up by the excitement of the little discoveries that you make along the way as you're sort of searching towards your big goal. And by the fact that you never know when you repeat the experiment if it's going to work out the same way, because sometimes things are different and the unexpected happens. And what's magic about that is that's usually telling you something new if something different happens. If your experiment is not the same as the one that happens on the other side of the world, something is different in the way you're doing it, and that something can tell you something new. So it's a terrific intellectual challenge. And the other thing is, sometimes artists say, well, you know, science, you don't have to be creative or imaginative. Nothing could be further from the truth. You have to have many wild ideas. Since this is Oxford, I'd like to quote Lewis Carroll, because the White Knight, if you remember, said, I try to believe 6 impossible things before breakfast. And I think that's a good idea for a scientist. You need to have masses of ideas, and you can be as imaginative as possible. You just have to remember that your ideas also have to fit the existing facts. Feynman put it very well. Science is imagination in a straitjacket. And so, what else am I going to say? Yeah, throw many balls into the air and hope to catch just 1 or 2. The other thing from my point of view of why science is so rewarding is you get many toys to play with. These wonderful machines and pieces of equipment that cost thousands, indeed sometimes millions of pounds, that you could never afford to set up on your own in the garage, and you get them for free. And as an academic scientist, being a scientist is a wonderful thing. Because another thing that you probably don't even think about is you have people working in your lab from all over the world. It's truly international. You've got collaborators all over the world. I've worked in Japan and Germany and the USA and Sweden and other places. And you get to travel all over the world to conferences. So, of course, if you're a zoologist or something, you end up in the Amazon basin or Antarctica, or studying lemurs in Madagascar or malaria in Vietnam. And that's not what I do. I'm sitting at a microscope, or these days a desk. But conferences are held all over the world. So I have explored Europe through the European Association for the Study of Diabetes. And it's a very social experience. Don't imagine that you'll be sitting there on your own, beavering away. We work in teams. And it's really rewarding. And the team, if the team is working well, then they also work well outside the lab as well. My lab, for example, just to give you two examples, one is they all ran in the Oxford Town and Gown fun race, which I strongly condone, since working on diabetes, I think the mantra is eat less, exercise more. And they also made a wonderful video film recently for when one of the students left the lab. And I think another thing about being an academic scientist is that you're your own boss, right from the word go, right from being a PhD student. You do what you want, when you want. And if you decide that it's an incredibly diverse life. So you teach, you give lectures like this, talks like this, you do science, you write books, you can do just about anything. And if you're going to be a scientist, I would say just a couple of words of advice. One is try to find your own niche. Don't copy what everybody else does. Don't jump into the same field because it happens to be a hot field. Because many other people will be working there and you can easily get burnt. My advice would be find your own area of research and be the expert in that. And then you'll find that everybody else will pile into it because you've made it so exciting. And have courage to try something new. Somebody once said, I think it was a guy called John Shedd, a ship in the harbour is safe, but that's not what ships are for. So go out there and try something new. And remember, science provides you with many rewards, not just the exhilaration of discovery and all the wonderful people who you collaborate and work with, some of whom I've been working with for 30 years. By the way, I should say, pick your collaborators carefully. I always choose mine on the basis of, would I like to go out for a drink in a pub with them? and if you work in the medical sciences like I do, you never know, you might be as lucky as me and be able to help other people. Because those are the real rewards, not the prizes that you win. It's seeing the kids in front of you get better. But remember, that's not why we do it. You can't sustain a life in science by imagining that you're going to go out and cure diabetes, for example. If you want to do that, you should be a doctor. You're not in the right job. You have to be in science because you're curious and you want to know how something works. You do it for the joy of doing it. Max Perutz said of Dorothy Hodgkin, who's our only female Nobel Prize winner in science, she pursued her crystallographic studies. She actually got the crystal structure of insulin, not for the sake of honours, but because that was what she wanted to do. So, since we're in a bookshop, I'm going to quote somebody else as well. Have I got time?
Yeah.
And that is T.S. Eliot, who is one of my favourite poets. And he has this marvellous line, which I used to say, which was, so here I am in the middle way, having had 20 years largely wasted. And he goes on to describe how it's a constant struggle to find the right word and to find a new way of putting it and to find the right emotion and the way to describe it, and how even when he finds he's done it, somebody else has got there before him and has done it better. Well, that might be poetry, and it might just be Eliot being morose and reflective on that day. But in science, it's not like that. Whatever you do, and however small a contribution you make, you are always building the wall. You are always putting a brick in the wall with your own contribution that enables other people to stand higher up and see further. So consider science as a career. Oh boy. I think that's a problem about Oxford. People ask all these difficult to answer questions. Well, you get my definition of science. I think science is trying to understand how the world works. That would be my definition. And anything which goes towards that, I would consider science. So, well, the reason I started to write the book, which you may know from having read the first one, life at the extremes was because it was a competition and it was sheer greed. The prize was £25,000, that's a lot of money. And I decided that I wanted that prize, so I would write the book. So it was for a life scientist who was practicing as a life scientist to write a popular science book. So I had a go, and I didn't win the prize, but the person who was the publisher on the committee said that he liked the book and he wanted me to write the book, and he'd offer me £17,000 to do it. So of course I did it. And to my amazement, it's done really well. It's sold over 100,000 copies. It's been translated into 12 languages. So I've been very happy about that. So I wrote it on Sunday afternoons. It took 3 years. And Life and the Spark of Life, that's about my own area. And I always kind of wanted to write something about that. Because actually what I discovered when I was writing the science book was that it's a kind of liberation. It's a totally different type of writing. Writing a scientific paper is very formulaic. You know, you write the methods, then you write the results, then you think about what it means and write the discussion. Finally, you write the introduction. So it's never done the way it reads. But you can be much sort of, it's a much more insecure form of writing because you open yourself up to people in a very different way, and so it's much more scary. But it's also, I found it challenging and fun, and I enjoyed it. But it was jolly hard work. You know, when I'd written the first chapter, I thought, well, that's nice, that's done, and then I thought, my God, I've got 8 more. So, oh, masses of them. I mean, we've published about 250 papers, so it's you can see that we, well, that's not just me, that's me and the team, you know. I think you should also think that I may be standing here talking to you, but the people who actually did 90% of the work, I did some of it myself at the beginning, are the postdocs and graduate students and the other people who work in the team, who are all brilliant and are now off running their own labs or doing different things. So little discoveries along the way. Well, for example, what we wanted to know was, I'll tell you a funny story. So we started out and we found that glucose closed the channel, right? Closed the pore. So the question was, we knew that glucose had to be broken down and metabolized to close the pore. So the question is, what was that metabolite? So I was pretty certain it was a chemical called ATP, which is normally used as the energy currency of the cell, and shouldn't be a kind of second messenger like this at all. And so that's what I looked for. And I was really pleased when I discovered this indeed happened. And I was not at all pleased when I then discovered that somebody else a few weeks later published a paper which said exactly the same thing. And I didn't get there first. And so this also happens in science. You have to expect, other people are working on it. And you have two choices, in that case. What you can do is you can say, I hate them, they're my competition. Or you can say, well, they're working on the same thing, why don't we collaborate? And that's happened to me. Sometimes they compete, and sometimes we collaborate, and the collaboration's been wonderful. But then we wanted to know what's the DNA sequence for the channel, how is it put together, how does the protein sequence relate to the functioning of the channel, where do the ions go through, where does the ATP bind, what causes this thing to open and close, all of these questions. The big question I'm after now is, what does it look like? We want the 3-dimensional crystal structure of this channel, and we've been working on it for at least 8 years. And since everything in my lab seems to take 10 years for a major achievement, I was sort of hoping another 2 years might be all it takes. But my colleague over there, who's smiling wryly, who is a crystallographer, will tell you that it's very likely it's going to take me another 10 years. Nothing at all.
Why not? Or they just care about animals? That sounds like a pretty scary thing to ask somebody to do.
Well, the thing is that actually it works like this, because the drug was in routine clinical use to treat type 2 diabetes. It had just never been thought of for this rare form of diabetes. They were supposed to have, it was thought they had type 1 diabetes, where you don't have any beta cells, so they automatically gave them insulin. They'd never tried the drugs. The drugs had gone through clinical trials 50 years ago, if one bothered to go through clinical trials in those days, and they had been used for many years very successfully. We just took them from adults and tried it out in babies as well as adults. And amazingly enough, you're allowed to do that without anything. So all it required was the consent of the patient and their physician. If we had had to find a new drug, I wouldn't be telling you this nice story now. It would be 20 years in the future. Cystic fibrosis is a very common disease, a lung disease, which is probably the commonest genetic disease in the UK. And they discovered the protein that was defective, the gene that was defective, probably 20 years ago, 25 years ago, I think. There's still no cure except for one rare form. That's because they have to find a new drug, and it takes a long time. So, yeah, we were very lucky. Many of our patients ended up in hospital. That's how they were found. Usually, what you find is that a child is failing to thrive. They're not putting on weight because they haven't got any insulin, which is a growth factor. They are crying. They're constantly peeing, and being sick and so on. So, and sometimes they end up in hospital with diabetic ketoacidosis. So, and then they measure their blood sugar, and then it's a full emergency. But I have a butterfly mind and one that never throws anything away, so I just collect stories. You see, I think telling stories is fun. I like telling stories. So I like being a storyteller. So this one I started because, of course, I taught that to my medical undergraduates when I was a medical tutor. I taught altitude physiology, what happens when you go into space, exercise physiology, all that sort of stuff. So I just used that knowledge and added the stories. And stories of, getting frostbite everywhere in the press. And there are many more that have come up since, many of the records here in this book, which is 10 years old, have now been broken. And with Life with the Spark of Life, of course, I knew many of those. And I did write a textbook before that on channelopathies, on diseases which are caused by ion channel dysfunction. No, not really. I only know those very early experiments. The Miller experiment. You know the one I mean? For everybody else, it's where you take a whole pile of basic stuff like nitrogen and so on, and you put it in a gas jar and you discharge electricity through it, and you can build some amino acids, things like that, basic building blocks, or other things. For me, I'll tell you what I think is really interesting. I'm fascinated by the fact that all the cells in our body, and just about every organism I can find, have as their main intracellular ion, cation. I always err on the positive side. Potassium. And potassium, it's really interesting that we have potassium inside us. Because if you ask people where life originated, they'll tell you it's in the sea. And the sea is full of sodium, not potassium. So I was wondering where on earth all the potassium came from. Why should we have potassium inside ourselves? We have sodium in our bloodstream. And that's understandable because cells got together in the sea and built multicellular organisms in the sea. So we have sodium in our bloodstream. But potassium, that's really weird. And so I asked various geophysicists and geologists about were the early seas high in potassium, and they all said, oh no, they were actually higher in sodium than they are today. So it's still a mystery. So I think, as I say in this book, I would go with Darwin, that life evolved in a warm little pond, probably amongst the clays, which are full of potassium, which could have leached out. And that's probably where the lipids first formed the first cell. But, that's huge speculation. I have absolutely no evidence.